science friends, let's review a little bit of cellular respiration with a few questions here. These are a little bit more challenging than most of our warm-up questions. So see if you can answer these. Why does NADH have more energy than FADH2? What you should answer here is that NADH drops off at a higher level in the electron transport chain. So we see NADH here, and we don't see FADH2 until down here a little bit later. And so it drops off at a higher level at the electron transport chain. And so these electrons are being moved further down the chain and longer down the chain, and so they're able to generate more energy to pump more hydrogens across, and more hydrogens represents more ATP that can be made, okay? And that brings us into our second question. Explain the idea of energy coupling that occurs in chemiosmosis. And so going back to this diagram, energy coupling is how hydrogen ions are pumped on this side. This is the matrix of the mitochondria. This is the intermembrane space of the mitochondria. And so down here we have high hydrogen ion concentration. Up here we have low hydrogen ion concentration. I know it doesn't really look like that in the picture, but that's how it is. Okay, they're just not all drawn here in the picture. And so essentially what this movement represents is its active transport, okay? So which means it requires energy. And so the energy is coming from these electrons being moved down the chain. Those electrons represent potential energy. And so that movement provides the energy that's needed to push the hydrogens across to the other side. Okay, and then we're going to build up a concentration gradient, all right? And so as we do that, we're going to build up high concentration of hydrogen over here. We flipped the gradient, and that's going to cause the hydrogens to want to move to the other side. And so they're going to do that through ATP synthase, which again is going to generate ATP through the spinning of this turbine, which is going to convert ADP into ATP, all right? And so this is energy coupling because we are using an endergonic process. This means it's absorbing energy, and the energy is coming from those electrons moving down the electron transport chain to drive an exergonic process. Exergonic meaning it's releasing energy, and again, that's in the form of the ATP here. Okay, so we're moving an endergonic process to create this concentration gradient up here at the top, and then drive an exergonic process, which makes ATP. And so that's the idea of energy coupling in chemiosmosis. In the next question, what element or atom helps to pull electrons down the electron transport chain? Maybe a better way that you would know that answer is if I ask you, what's the electron acceptor that helps to grab those electrons at the end of the chain? And so you should say that's oxygen. Okay. And then the last one, how many total ATPs are produced per molecule of glucose in aerobic respiration? And as we mentioned yesterday, it's not an exact number. This is saying about 30 to 32. I read something just a moment ago that said 38 ATPs. And so we're going to go with somewhere in the 30 range. As long as you know that it's like 30-ish, not like 100, then we'll consider that good enough. Okay. Remember that we know that there are 2 in glycolysis. We know that there are 2 in the citric acid cycle. This number is a little bit more variable, the oxidative phosphorylation. Okay. So that is that for review. So let's jump in today to alternatives to aerobic respiration. And so remember, we said aerobic respiration was with oxygen. But sometimes oxygen's not available. Sometimes there is no oxygen. You run out of oxygen. And so in that case, organisms can use one of 2 different processes. And so they may use a process called anaerobic respiration. And so in anaerobic respiration, they still do all 4 steps. the glycolysis, pyruvate oxidation, Krebs cycle, and the electron transport chain. But the difference is the electron transport chain has a different final electron acceptor. So it doesn't use oxygen as the final electron acceptor because there is no oxygen in anaerobic. And so what oftentimes it uses instead is sulfate. You may recognize sulfate from chemistry. And this happens in some types of bacteria. Okay, now not bacteria like the kind that we're familiar with that, are on our hands and that type of stuff. These are bacteria that often dwell in marine environments. They are in mud marshes and stuff like that. But they use sulfate as the final electron acceptor. And in this process, one of the products that's produced is hydrogen sulfide. And hydrogen sulfide produces a rotten egg type smell. And so in these cases for these bacteria in these environments, again, not, really things that we have around here, not in any environments that we have around here, you would recognize that rotten egg smell from anaerobic respiration. Okay, so that's one thing that can happen. But what I want to focus on today is the other option, which is fermentation. So in fermentation, there is no electron transport chain. Okay, furthermore, there is no Krebs cycle. Instead, we have glycolysis, and then a fermentation process results. Okay, so there's lots of different types of fermentation that you can have, but today we're going to talk about two main types, alcoholic and lactic acid. And they are named by the product that is formed. So in alcoholic fermentation, we form an alcohol, specifically ethanol, and in lactic acid, we form lactic acid. Okay, and so we're going to talk about those going forth. So let's start first with alcoholic fermentation. So in alcoholic fermentation, all of this process is going to happen in the cytoplasm. We are not going to move forward into the mitochondria. So fermentation is going to take place in the cytoplasm, as we see here. So this is our aerobic is over here. Anaerobic or fermentation is over here. And so you see that it's all happening in the cytoplasm. Okay, so what we had our glucose molecule, and we did glycolysis, and glucose was converted into 2 pyruvates, like we've seen. Okay, we released ATP in the process. 2 ATP are made in glycolysis, and as we broke down that glucose, we released electrons, and so those electrons were picked up by NAD to form NADH. All right, and then normally those NADH would go to the electron transport chain, but in fermentation, we don't have the electron transport chain because there's no oxygen to accept the electrons in the electron transport chain. So we talked about that a little bit yesterday, right? So normally we've always had NADH and FADH2. And every diagram we drew, we said to the electron transport chain. Those two go to the electron transport chain, right? And so normally NADH and FADH2 drop off those electrons and become NAD and FAD. And those electrons move down the electron transport chain. And at the end of the chain, they are given to hydrogen. Oxygen. Okay, oxygen. But what happens if there is no oxygen there ready to grab those electrons when they come down? Well, they don't just fall on the floor. Okay, so instead what happens is this process backs up. So NADH and FADH2 can't drop off the electrons, okay? And so what we're trying to do in fermentation is recycle NADH and FADH2. And that's what's happening here. You see in this diagram, these are being recycled. And so we're getting new electron acceptors, okay, to free up these NADH and FADH2 so that they can go back down and break up just a little bit more glucose so that we can get just a little bit more ATP. Now, we're not going to get all that ATP that we usually get in the electron transport chain, and even that little bit of extra ATP that we normally get in the Krebs cycle, because we're skipping those steps. We can't do those steps without oxygen. okay? But we're going to try to get squeeze just as much as we can by continuing to break down the sugars, glucose through glycolysis. So we need to free up these NADHs to be able to do that, okay? So back to this diagram. We've got glucose. In the process of glycolysis, it's being broken down into two pyruvates. So we have our pyruvate molecule right here, okay? Pyruvate is going to be converted into acetaldehyde, okay? Aldehyde was one of the functional groups that you learned at the beginning of the year. This is an aldehyde, CH with a double bond to an O, okay? That is an aldehyde. Now remember, in aerobic respiration, in this process, the pyruvate had an enzyme added to it called coenzyme A. That's in aerobic respiration, not in this step. And it made a molecule called acetyl-CoA. But you might recognize this also starts with acetyl because they're very similar in structure, okay? It was acetyl-CoA. This is acetaldehyde. And so structurally, when you look at pyruvate right here, and when you look at acetyl-CoA, everything is basically the same, minus this carbon dioxide, which was released. This tells us here that CO2 is a byproduct. And then the hydrogen was added from NADH. It is the electron acceptor, so it gained those electrons. It also gained the hydrogen and converted back to NAD+. So now NAD+ can be recycled and pick up some more electrons in glycolysis, okay? And so once this molecule, acetaldehyde, is then changed into ethanol, okay? And so when you look structurally at ethanol, it's also very similar, again, with just the additions of the two H's from NADH to NAD, okay? And so ethanol is that product of alcoholic fermentation. It has an alcohol functional group on it. If you think back to your functional groups, OH is sometimes called a hydroxide. You may remember that from biochemistry. And that's they're frequently found in alcohols. Alcohols often end in OL, okay, like ethanol. And so that is our product here in alcoholic fermentation. Alcoholic fermentation is also used a lot by bacteria, specifically a bacteria that you are probably familiar with, which is yeast. Alcoholic fermentation is used in brewing, in making beer, and making wine, and making other liquors. It's also used in baking, in making bread. If any of your moms or grandmothers make bread, they'll add yeast and sugar to feed the yeast. And that bread rises, right? And so what causes the bread to rise? Well, it's the CO2 that produces, okay? Sometimes if you open a loaf of bread, your bread will have like holes in it. Like it looks like, just a hole in your bread. And that bread is, that was a CO2 pocket that was trapped in the bread from when the yeast did alcoholic fermentation and to be able to break down that glucose and produced CO2 as a byproduct. Okay? So this is really important, used a lot in foods. All right? And so again, the goal is just to be able to regenerate this NAD to allow for a little bit more ATP to be produced. But all the ATP we get here is those 2 ATP from glycolysis. All right. Now, let's look at lactic acid fermentation. In lactic acid fermentation, we have glucose is converted to pyruvate, which is converted to lactate. Okay, now lactate is an ionized, or lactic acid is an ionized form of lactate. And so glucose is broken down. We've got those 2 ATPs released, okay, converted to our pyruvates here. And then these pyruvate is going to be our electron acceptor. So it's going to accept the electrons. Right here is going to be 2 electrons that make up that bond, okay? and then there's going to be 2 electrons that make up that bond. And so when you look at this, we've added on those 2 H's from the NADH. Structurally, those 2 are the same. And so in this process, we convert pyruvate to lactate, okay? And this process is called lactic acid fermentation. And so this happens in muscle cells. When oxygen is not present, your muscles opt to do lactic acid fermentation. And that lactate that builds up is thought to be a lot of what causes like muscle cramps and muscle soreness after a really strenuous exercise. The following day, you'll often experience a lot of muscle cramps and soreness, and that's thought to be from lactic acid fermentation. So we have our pyruvate that was reduced by NADH, okay, means the pyruvate gained electrons if it was reduced to form lactate. Now, one thing that's important about this one is that there's no CO2 released. In the alcoholic fermentation, we had this CO2 that was released in the conversion of pyruvate, but you don't see that taking place in the lactic acid fermentation. Okay, so that's a major difference there. All right. The last thing I want to talk about is obligate anaerobes and facultative anaerobes. So I want you to look first at this word obligate. When you see the word obligate, it should make you think of obligated. So if you are obligated to do something, maybe you're obligated to go to your little cousin's birthday party. What does that mean? Well, it means like you kind of feel like you have to do it, right? You basically have to go. And so obligate anaerobes mean they have to be anaerobic, okay? They have to do anaerobic respiration or fermentation. They do not do aerobic respiration. They cannot survive in the presence of oxygen, okay? You may also have obligate aerobes that can't survive without oxygen. We are obligate aerobes. We cannot make it without oxygen, okay? Not, beyond a couple of minutes anyway, okay? And so that's what that word means. Obligate aerobe or obligate anaerobe mean they have to have with or without oxygen, whichever way. Now we have another word, facultative, okay? And that word is kind of derived from like facilitate. And so they can facilitate aerobic or anaerobic, and so they can survive either way. They can alternate between each one. And so certain of our cells, like our muscle cells, can do this. We can use aerobic respiration. That's generally what our muscle cells prefer to use when oxygen is available. It gives us more energy, more ATP. But if oxygen's not available, they'll switch over to that lactic acid fermentation and do that step, okay? And so facultative anaerobes, that's what that means, with or without, survive using fermentation or aerobic respiration, okay? Now, one thing that's not listed here in the slides specifically, but one thing I want to talk about is kind of how evolution ties into all of this. So we talked about in the beginning in our very first unit that evolution is kind of a overarching theme that you see throughout the course of biology. And so evolutionarily, which process do you think started first? Aerobic respiration or anaerobic respiration? Think about that for a moment. Hopefully you said anaerobic respiration, okay? Anaerobic respiration is likely the oldest form of respiration. For one, it only happens in the cytoplasm, okay? It only, or fermentation, if you want to say fermentation or anaerobic, either of those would be okay. It only happens in the cytoplasm. And so we know that prokaryotic cells are older than eukaryotic cells. Eukaryotic cells don't have that mitochondria. And so probably the source that prokaryotic cells use, and now they can do aerobic respiration in the cytoplasm. We talked about that. Prokaryotic cells do aerobic respiration in the cytoplasm. But anaerobic respiration is probably the oldest form. And I just also want you to see that this is kind of a theme that ties all things together. Both both prokaryotes and eukaryotes use glycolysis. And so that is something that's kind of similar to the two. Most scientists believe that, two things wouldn't develop these processes independently, okay? Rather, they probably both were one thing. They both had a common ancestor that did glycolysis and then split off to two different pathways beyond that. Okay, so that's all I want to say about that today. And we'll go into more about evolution and all of that type of stuff later on. So if you all have any questions, then let me know. Other than that, that's all I have for anaerobic respiration and fermentation today. And you're going to finish up your worksheet on respiration. You're going to do Model 3 today. I think I told you yesterday in the lesson you were doing Model 3, but actually you were doing Model 2 yesterday. Today you're going to do Model 3 and submit that. And then tomorrow we will move on to photosynthesis. So I hope you all have a great day. Let me know if I can help with anything. Bye.